Chào sáng hảo, xie sân man. Good morning, welcome, welcome. Hello, hello. Okay, this is 9C, and there are 30 of you, right? Wait. Okay. Kamera saya hitam karena wifi saya sedang jelek. Oh, okay. I really hope that the connection of yours will be recovered very soon. Lavinia, okay. Thank you for informing me. Is that correct that there are 30 of you in your class, guys? Yes, okay. So that means along with Miss Lita and I, that makes 2 of us teachers, means there are 29 of you. Here comes Mr. Keenan. So there are 32 of us. I thank God. This means that all of us are here inside the meeting room. Thank you very much, everyone. Yeah, we are going to continue our activities, and yeah, this yes. Anybody want to say anything? Anybody want to say anything? Anyone? No? Okay. Yeah. Good morning again. Leader, do you want to greet us? Go ahead.
Greetings.
Good morning, sir.
Good morning, sir.
Yeah, morning teachers, because there is Miss Lita here, right? Would you please do that one more time?
Greetings.
Good morning, teachers.
Good morning, teachers.
Good morning, everyone. Good morning. By the way, who's the leader? I mean, it's not spotlighted. Who's the leader? Okay. Who's the class leader? Oh, Gerald, yeah. Gerald, right? Oh, yeah. I mean, your voice is deep, yeah? Okay, good. Okay, everyone, I want you to go to Nearpod now. Because we are still in the middle of our interaction and discussion related to the reproduction of living organisms. We have been talking about the plants so far. Not yet the animals. I really hope that very soon we would be arriving at the discussion about animals, but I'm going to continue. We are going to talk about something important first, which is, I think, the fundamental matter, okay, related to this. So I'm gonna ask you to go again to join.nearpod.com. I'm sorry, join.nearpod.com, and I'm going to share the link, and I'm going to paste the link in the chat. This should be directed to everyone in the meeting, and here it is. Yeah, go ahead, guys. Please come on in, enter the meeting room. We've been talking about the reproductive system, and I mean, not really reproductive system, but reproduction in plants, right? And we've been talking for like several sessions, but there is something you need to understand. I mean, something we need to understand as we are digging deeper into this topic, especially when we talk about the reproduction and angiosperms. Okay, which is, already, you've been asked to read that there is what's so-called the double fertilization, things like that. About the number of nuclei along the process. Yeah. So for this purpose, I mean, our understanding about mitosis, meiosis, which are the cell divisions. I noticed that there are only 7 of you yet, or 30 now. Oh, remember that whenever there is something wrong with my network, please inform me, okay? Immediately, so that we can find the solution immediately, because it's important, very, very important. Okay. I'm going to share my screen now. I'm going to show you this. Yep. Okay. So mitosis, meiosis. It's been already talked about a little bit before, but have you really understood about these concepts? Okay. Two important concepts which will explain to you about the number of nuclei involved in the process. So let's get on with this. Yeah. It's like we are going stopping at a pit stop, and then we pick up something which is very good for our next race in understanding this topic. So 23 of you here inside. Come on, everyone. 27. Join. Because why? Because there are questions, things, activities in the near part in which you will be assessed as well. I mean, I'm going to look at the score you are able to achieve in all the activities, and that will be considered as something which hopefully can add up or contribute in strengthening your overall grade. Okay, overall grade. So go ahead, guys. There are 29 of you, so I believe there is one more. Give me the strings for one more. Okay, we are going to start. Okay. Let me see what we have on. Oh, it's freezing. Okay. Carry on. I hit the next button. Hopefully it works fine. Come on, button. You okay there? Oh, there you are. So the learning outcomes. Still, the learning outcomes. One of the learning outcome, which is the main one, is to understand plant and animals reproduction, right? And the second one, especially today, I want all of us, including me, to deeper understand cell division as one fundamental concept required in this chapter, in understanding this chapter. So the second learning outcome is specifically chosen for today's activity. Okay. Now, first we are going to talk about mitosis. Okay, we're going to talk about mitosis. I haven't made any song related to this. I really hope that I can, but recently I feel that it's like 24 hours per day is not enough. Okay, so I got a video. Okay, I'm going to play this video, and I hope that you can participate in this video. I will allow you to play it, and remember that you have to participate in all the activities inside. Go ahead. I'm going to allow you to play the video individually while I play it here.
Have you ever looked down at your leg or your arm to find some cut and you have no idea where it came from or how you got it? So you put a bandage on it and a few days later it's gone. It's all healed and you don't even think anything of it, of this amazing process that causes this to happen. Or let's say you're looking at your nails and you notice they're a lot longer since the last time you cut them. Or let's say you're looking in the mirror and you notice that you are a lot bigger than you were when you were 5 years old. What do all of these things have in common? Well, one major thing they have in common is mitosis. Mitosis is a type of cell division done by most of your body cells, and it's really important for cells to divide. If they didn't divide, you wouldn't grow. I mean, how do you grow if you can't make more cells, right? So one reason why you're bigger than you were when you were 5 is mitosis. Mitosis also is great for repair of damage. If you have some kind of accident, like when we were talking about that cut on your arm or leg, well, you want to make sure it gets repaired well. So you have to make more cells to do that. Mitosis is great for that. Now, it's really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells, because that's something different called meiosis, which sounds like mitosis, unfortunately, but it's a different process. Mitosis is done to produce body cells. Mitosis makes identical cells. That is the goal, identical cells. So if you're trying to make more skin cells to replace worn out or damaged skin cells, you don't want to start suddenly making stomach cells there. That would be ridiculous. You want to make sure you have identical cells replacing what was lost. So mitosis makes identical cells. It's a really important thing.
Yeah, there is a activity in the form of multiple question, multiple choice question. I mean, go ahead. Which one is the thing that mitosis doesn't do? Which one? I mean, as this time from this moment, from this second onward, you really need to pay attention on the content or what the video is saying, okay, so that you can answer all the questions. I noticed that many of you already answered. Yeah, it's supposed to be quite easy, right? It's supposed to be quite easy. What mitosis doesn't do is, okay, great. I'm going to continue then.
Now, it's also important to understand that your cells are not dividing all the time. If all they did was divide, it would just be rapid, crazy growth. In fact, this is kind of what cancer is. Cancer is uncontrolled cell growth.
What is cancer? What is cancer? Cancer is what? Yeah. What is cancer? Yeah, okay. What is cancer? Cancer is not part of a horoscope, right? My zodiac is Cancer. No, not that one. This is something that take place in the body of organisms, yeah, especially when we talk about human beings. So what is cancer? Cancer juga bukan kantong kering, okay? Carry on. Continue to answer. Participation 72%. I really hope that you could answer all the questions, because at the end, this is graded. Okay, guys, this is graded. So what is cancer? That is the question right now. What is cancer? It was just mentioned in the video what cancer is, right? And yeah, I guess most of you already answered this. Participation 83%. That's awesome. Come on, no, I'm not really going to be waiting for 100% at 97. Oh, nice, awesome, guys. I'm going to continue then. I'm while still waiting for you
We have a
maybe
clip
one or
on
two
the
of
cell
you.
cycle and what the cell is usually doing most of its daily life, which is actually a phase called interphase, where it's growing and replicating its DNA and carrying out its daily cell functions. That's where cells spend most of their time in respect to the whole cell cycle. Mitosis is a very short amount of time in respect to the whole cell cycle. But mitosis is a critical process because this is where it is going to divide and make more cells.
Yeah, interphase alone constitutes most of the cell cycle, up to 90% of the whole cycle. What are the three things occur during interphase? What are the three things? Means here you need to select the, well, okay, if I do this, I mean, you will be what if I collapse this? No, I don't want to do anything which potentially not good. Okay, guys. Okay. 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 Yeah, it's pity that some of you got no answer for this one. Yeah, pity. Yeah. So because you really you really out to have that question. I mean, try to figure this out. Okay, try to figure this out. Okay, I'm going to continue then. 72, 21. Yeah, 7 no answer. Too bad. Carry on, carry on.
Before we get into the steps of division, it's really important to understand that your cells have something inside them, an organelle called the nucleus. And the nucleus holds your DNA. DNA is really important because it's your genetic information. And if you're going to make more cells, you need to have the same DNA in those new cells as you did in your original cells. You want it to be identical, no mistakes. Very important. The problem is you've got a lot of DNA, and we've got to get that DNA into the new cells using mitosis. So there has to be a better way to organize that DNA. Well, what actually happens is that DNA can be organized into these condensed units called chromosomes. Chromosomes are made of DNA and protein. You've probably heard before that humans have 46 chromosomes. That means 46 chromosomes are found in most human body cell nuclei. What are nuclei? Well, it's the plural of nucleus. You don't say nucleuses, you say nuclei. Well, in the nuclei, there are 46 chromosomes. Organizing DNA into condensed chromosomes makes it a lot easier to move over when you're making new cells. So if you have 46 chromosomes in a human body cell, you have to duplicate those chromosomes in interphase before mitosis starts. That basically means you're duplicating your DNA, since chromosomes are made of DNA and protein. You have to do this before mitosis starts because if you're going to make an identical cell that has 46 chromosomes, just like the original, well, it makes sense you have to duplicate the genetic material before splitting. So if you look at our cell cycle video clip, we talk about interphase. That's a stage where most of the time cells are spending their time. They're actually duplicating their DNA during that time. So ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, as the sister chromatids are still attached, and we're counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture, but you went from 46 to 92 chromatids. We have a video.
Okay. I really hope that you understand that part just now. Yeah. So strands of replicated chromosomes is called which one? Is it centromere or chromatids? Yeah. Hopefully this is an easy question as well. Okay. Okay. Now in this session you really need to focus. Okay, guys. Awesome. Yep. Oh, come on. Okay. Then I'll continue, guys. I'll continue then.
I have a video explaining that
Yeah, I
in
guess
more
that's it.
depth and how that factors in for mitosis. Okay, so now we can get right into mitosis. I like to tell students to remember PMAT. It's a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the A is for anaphase, and the T is for telophase. So remember PMAT, the stages in order. The very first step is prophase. Prophase because it's the beginning step, the nucleus is still there, and it's going to...
Yeah, what is the correct order of mitosis phases? Which one is the correct order? Is it A, B, C, or D? Okay, it's mentioned before, and it's supposed to be, yeah, it's supposed to be easy. I hope that yeah, there are still no answer. Okay, contributed here in this activities. Yeah, unfortunately. Okay, so it's what? PMAT, yeah, pro, meta, and telo. Okay, very good. Carry on.
It's going to go away later on, but this is a stage where it's actually still there. The chromosomes are visible. In fact, we say they're condensing, which means they are thickening and visible. The next stage is metaphase. M for metaphase, but I also like to remember M for middle because in this stage, the chromosomes line up in the middle of the cell. The nucleus has been disassembled, it's no longer there, so we've got the chromosomes in the middle waiting there. Next, the A is for anaphase. In anaphase, I like to think as the A for away. The chromosomes move away. They are moving to opposite sides of the cell, so they are moving towards the poles of the cell. Now, one thing to point out, these chromosomes, they're not moving by themselves. They actually have something called spindles. These spindles are fibers that help move the chromosomes to the ends. Kind of helps them move along. The last...
Yeah, what dragged the chromosomes from the center, the place where they was lining up, okay, and then it's dragged by this thing, the first item, and then it will be dragged toward where? That would be the second item. Okay, something drag, the thing that drag, and then the destination. Okay, A, B, C, D, spindle, centriole, centrosome, centriole, spindle, centrosome, centriole, centrosome. Which one is the correct one? And 90 got this correct. No answer is still there, unfortunately. Too bad. Oh my gosh. Okay. Yes. Okay, thank you very much, everyone. Let's carry on.
The last stage of mitosis, think T for telophase. In telophase, the chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make these two new cells. The nuclei are starting to surround the chromosomes on both sides. I like to think of the T for 2 because you can really see in this step that the end goal is going to be 2 cells, and in the human body, they're each going to have 46 chromosomes. And again, remember, they are identical. Now, cytokinesis is responsible for the final separation into 2 cells by splitting the cytoplasm, which completes after the PMAT mitosis stages. So why did all this matter?
What is cytokinesis? What is cytokinesis? What is cytokinesis? Cytokinesis bahasa Indonesianya. Apa sih cytokinesis itu? Yeah, based on what you heard just now in the video, I really hope you could capture that definition just now and then type it here as your answer. What is cytokinesis? Has there any participation? 31%. Nice. Very awesome. Yeah. The process of, until there you got this correct. Okay, cytokinesis. What is cytokinesis? And yes, actually this is the last part after all the internal interesting, not really simple processes going on, then this one, cytokinesis, need to happen, because that's actually the idea of doing mitosis in the first place, right? So what is cytokinesis? What is cytokinesis? What is cytokinesis? Go ahead, go ahead. It's time for you to answer this after you catch the definition shared in the video just now. But I don't think I'm going to be too long here, even though participation is still 79. I cannot wait for all of you. Hopefully you would be able, you could answer this ASAP. So when I leave, okay, I'll just leave. It's okay, because you can play the.
Without understanding cell division, we wouldn't understand how growth and repair happens, because they both require more cells to be made. Understanding mitosis is also very important for cancer research. Cancer itself is uncontrolled cell growth. So, in other words, uncontrolled mitosis. Well, that's it for the Amoeba Sisters,
Yeah.
and we remind you to stay curious.
Stay curious, even though we know what the saying is, curiosity kills the cat, but I guess being curious is very good. Okay, yeah, I'm going to continue then. Thank you very much for your participation in this activity. Awesome. Now, after we talk about the mitosis, and you also watched just now that the way these amoeba sisters try to enable us to easily remember what happens during the PMAT, each and every one of them, PMAT, Prophase, Metaphase, Anaphase, Telophase, is that they use the initial to represent something, okay? Prophase, pro means before, so before everything happens, the processes take place, pro, and then metaphase, middle, means at that time all the chromosomes are lining up at the center and in the middle of the area, the equatorial area. And then anaphase, stands for, she said, amoeba sister said, away, because the chromosomes like moving away from each other, right? Away. And then yeah, this is an interesting way of understanding this. I really hope that you all would understand this concept, the previous one. Now we are going to get on with the second item we are going to talk about today, which is meiosis. Okay, let's watch the video. I'll allow you to have your own pace in watching this. And again, don't forget to do the activities, okay? Amoeba sisters. Meiosis.
Have you ever wondered how two siblings can have the same mom and dad and still look so different? Well, today we're going to talk about a process that makes that possible, a process called meiosis, not to be confused with mitosis, which sounds unfortunately similar. Mitosis makes identical body cells, like your skin cells and stomach cells. Recall from our mitosis clip that since it makes identical body cells, mitosis is important for growth and for repair of damage, or to replace worn-out cells. But not meiosis. Meiosis is a process that contributes to genetic variety. Meiosis also doesn't make body cells. It makes sperm and egg cells, otherwise known as gametes, the fancier word. You might recall that humans have 46 chromosomes. That's how many chromosomes most body cells in your body have. But there are some human cells that don't have 46 chromosomes. Human sperm cells and egg cells have 23 chromosomes. Why the number difference? Well, if a sperm cell has 23 chromosomes and an egg cell has 23 chromosomes, when they come together, that makes 46 chromosomes. That will allow a newly formed fertilized egg to develop into a human. Meiosis is what we call a reduction division because you have a starting cell that has 46 chromosomes. And your ending cell, the sperm and egg cells, have only 23 chromosomes. Before we can start getting into the stages of meiosis to make gametes, we have to remember what happens before meiosis can even start. Actually, this also happens before mitosis. It's the stage known as interphase. And if you're
The following are things occur during interphase, except actually this is kind of deja vu, okay? Deja vu. So I really hope that you will be able, you can answer this question. Piece of cake, Lee. This will be so very, very easy, okay? Because it's a deja vu. You've seen that just now, less than half an hour ago. Of course, it's like several minutes ago. Great, awesome. 96 correct, no answer for. Oh yeah, 100% correct. Of course, you guys are smart. I know it.
And if you remember interphase, it's when the cell is growing, it's replicating its DNA, it's carrying out cell processes, and just like mitosis, interphase happens before meiosis is even going to start. So the starting cell has 46 chromosomes, and you have to duplicate those chromosomes in interphase before meiosis starts. That basically means you are duplicating your DNA, since chromosomes are made of DNA and protein. Ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, because the sister chromatids are still attached, and we are actually counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture. But you went from 46 to 92 chromatids. Little tricky there. We have a detailed video that explains these chromosome numbers before and after replicating in interphase that can be useful for meiosis. Okay, so interphase checklist done. Now we can move into meiosis. You might remember the mitosis stages PMAT. The P was for prophase, the M was for metaphase, the A for anaphase, and the T for telophase. And the good news is that in meiosis, you still use those terms. But because meiosis is actually a reduction division, you're going from 46 chromosomes to 23, which means you actually divide twice. So instead of mitosis, where you divide one time and do PMAT one time, in meiosis, you're going to divide twice and therefore do PMAT twice. And because
Why do you call meiosis reduction division? Why do you call it reduction division? It's explained just now, like twice. Okay, why do we call meiosis reduction division? Reduction means it's lessen. It's reduce, right? It reduce something. Something is reduced. Something is not as many as that thing before. So reduction division. Why do you call meiosis reduction division? Go ahead. I hope you were successfully able to caught the definition of this thing. Participation is still 29. Reduction division. Yeah, like I said before, it's a yeah, again, meiosis is a cell division, right? But the difference between meiosis and mitosis is that mitosis is not a reduction division, while meiosis is a reduction division, because at the end of the process of meiosis, in which the PMAT happens twice. The PMAT happens twice. Yeah, prophase, metaphase, anaphase, telophase. It happens twice in meiosis. At the end, the chromosome, the set of chromosome is different. I'm leaking the answer actually. So I'm gonna stop saying anything. Okay. So 46. Oh, seems like this is not a this is not really easy for many of you, right? So why do you call meiosis reduction division? Okay, 50% participation, 50% participation. Divides twice. Yeah, okay. The PMAT happens twice is not really the main thing that then differentiate meiosis to mitosis. Yeah. Yeah, because the 46 chromosomes become yes, good. Yeah. Okay. Reduces the number of chromosomes you have, right? Awesome. Yeah, divides twice. Yeah, divides twice again is not really the essence. Okay, it's not really the quintessential part of this. But what happened because of the twice division, that's what matters. Okay, good. Very good, everyone. So let's see, it's 79. Great, because PMAT happens. Typo. Okay, I understand. I mean, sometimes no, I mean many times I do typo. Okay, many times. Yeah, it's like my fingers are all thumbs. Okay, so I'm going to proceed. Okay, I'm going to proceed. I'm going to continue. 86. Will it be 100% participation? I really hope so, everyone. Nice. 93. Hallelujah. Good. I'm going to continue then. Let's go.
And because of this, in meiosis, you put numbers after the phases to indicate whether you're in the first division or the second division. So let's dive right in. So let's start with the very first step, prophase 1. One thing I like to remember about prophase is this actually means before, and it kind of helps you remember that it comes before all the other stages start. This is where the chromosomes are going to condense and thicken. They are actually going to line up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. They're going to match up. It is during this prophase 1 that this amazing process occurs called crossing over. Now, I know crossing over probably sounds like something very different, but this is actually a really awesome process because this is when the chromosomes, they're lined up in homologous pairs, and they have this way that they can transfer their genetic information and exchange it between each other. It's kind of like these chromosomes flop over each other, and they do a little genetic information exchange here. It makes for what we call recombinant chromosomes, which can eventually contribute to the variety that we were mentioning that siblings can have even when they have the same parents. More about that later. Now,
the exchange of genetic code between a pair of homologous chromosomes is called, what do you call the exchange of that genetic part? Bump to each other, and there is exchange like instantly happens. Okay, and the result of this occurrence is called what? Okay, just need to choose one out of two. Which one is the correct answer? The exchange of genetic code between a pair of homologous chromosomes is called what? That would be the first item in the option, and the result of this occurrence is called what? Second item. Okay. This is supposed to be easy. This is supposed to be easier. Easy like Sunday morning. Yeah. Okay, I'm going to continue then. Let's go.
Now we move into metaphase 1. In metaphase 1, think of the M as standing for middle. The chromosomes are now going to be in the middle of the cell. It's a little bit different, though, from mitosis because these chromosomes are going to be in pairs in the middle of the cell. So it's not a single file line. They're in pairs in the middle. Now, during anaphase 1, think of A for away because the chromosomes are going to be pulled away by the spindle fibers. Then we end with telophase 1, where you have two newly formed nuclei, and it becomes obvious that you're going to end meiosis 1 with two new cells. Cytokinesis follows with splitting the cytoplasm. But we're not done yet. On to meiosis 2. The very first step in meiosis 2 is prophase 2. It's not going to be nearly as eventful as it was in prophase 1, though, because they're not going to have homologous pairs of chromosomes. They also are not going to have this amazing process called crossing over. That doesn't happen again in prophase 2. You have your chromosomes, and the spindles are starting to form like they did in prophase 1, but prophase 2 is just not as eventful of having that process of crossing over. In Metaphase 2, remember, think M for
Middle.
middle, the chromosomes are going to line up in the middle. This time, though, they are in a single file line. They are not in pairs like they were in Metaphase 1. Anaphase 2, remember
Okay.
A for away, but this time it's the chromatids that are going to be pulled away by the spindle fibers. In Telophase 2, you can see the nuclei reforming, and you can also see that the two cells have divided. There's going to be 4 cells forming. Cytokinesis will follow to completely split the cytoplasm. Now, keep in mind that meiosis in males produces sperm cells, and in females it produces egg cells. Because of independent assortment and also crossing over, you're going to have variety. For example, in a male, the 4 sperm cells that are produced each time, they are all different from each other. They're also different from the starting cell because the starting cell had 46 chromosomes, and the ending cells only had 23. So they are not identical to the original, and they are not identical to each other. This is
So how would you attempt to memorize what happened during each of the 4 phases of mitosis or meiosis? How would you identify which phase is happening to a cell when we observe a cell under a microscope? If you look at a cell and you could see what happened in the nuclei, how could you tell that this one is undergoing P, or this one is undergoing M, or this one is undergoing A, or this one is undergoing T, or this one is undergoing Interphase? Okay, how would you memorize this? I mean, this test your understanding of the characteristics of each and every steps. Okay, yeah, I will hope you already got an idea that, okay, if I see that the chromosomes are already lining up at the center, at the middle, then that should be that process, PMAT, any which one you think it is. Okay, or now I could see that the chromosomes it's like they are still sister chromatids, right? And they are pulled away from each other, and this must be one of the PMAT. Remember, it could be meiosis 1 or meiosis meiosis 2. It could be something like that. So but in this case, I won't be forcing you to think of this as a question when you are focusing on the meiosis. So this should be general mitosis or meiosis, PMAT, how to differentiate them visually. 27. So this is not really an easy question to answer. I guess I'm going to skip this one. Okay, let's go then.
This is going to lead to variety, a reason why 2 siblings with the same parents can look different from each other. They still developed from a unique egg and a unique sperm cell that came together. One last thing to think about. Scientists are often looking into the process of meiosis because sometimes the chromosomes don't separate correctly. It's called non-disjunction when a cell can receive too many or too few chromosomes in the separation. This contributes to some genetic disorders, which is something scientists continue to study. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Yeah, okay. That's awesome. Mom says I can do my until I finish my interface. Yeah. That's awesome. Okay, guys. Yep. One out of 8. Yeah, this is another activity. I once what's that switch view, yeah, and that caused problem. So you are doing another activity right now, and I'm looking at the progress of it. So hopefully this could happen quickly enough. Yeah. 3 out of 8, 1 out of 8, 3, 8. Yeah. So again, this is a quiz which testing your memory, because before I ask you to read, and actually, even though I don't ask you to read your book, but reading the book bit by bit, any subject, especially biology, which many people think as it got so many so much materials. Yeah, it's important. Okay, read. So should I show name? Yeah, okay, it's a multiple choice. Remember, it's about the seed dispersal. Okay, one is about seed dispersal. One about the seed dispersal. And then the second part would be about the types of pollinations. Okay, can you able to answer this correctly? Oh yeah, okay. Oh, that one. Oh, number 4. Okay. Let me see. That's number 4. Yeah, okay. Is there any of you able to, finish this one, this quiz? Go ahead, guys, do the quiz. Let's see if I can change the view to student view. Changing to student view. And okay, we got only a little bit of time left. Okay, I cannot be here too long. So let's see what we have here. Oh yeah, 8-question quiz. I'm going to switch again to the teacher's view, and I'm going to close this. Okay, but one lesson for all of you: start to read. Okay, because none of you, I noticed, already answered 8 out of 8. Many got 7 out of 8. That's good, but not yet any of you answered 8 out of 8. So I'm going to continue, guys, because of the time. I'm going to continue. What I'm showing you here is a diagram. It's a table which tell us the difference between mitosis and meiosis. Please keep on reading about this, okay, guys. We know that both consist of PMAT, okay, prophase, metaphase, anaphase, telophase. It's just in mitosis there is only one cycle of PMAT, and in meiosis there are 2 cycles of PMAT. And what you need to understand is that the term haploid and diploid, okay, haploid and diploid. When we say diploid, that is our body cells. Our body cells consist of diploid set of chromosomes, means we got 46 chromosomes or 23 pairs of chromosomes. So all cells that undergo the 2 types of cell divisions all got 46 chromosomes. But see here that at the end, in mitosis, the mitosis will produce daughter cells which are also diploid, means having 46 chromosomes. Yeah, the symbol is 2n. Okay, 2n. While in meiosis, Before it was diploid to N, but at the end it will be only N, which is half the set of the mother cell. Okay, so the daughter cells are haploid, so they only got 23 chromosomes instead of 46. Okay, I guess that's it. Please read. I'm going to share with you the link so that you can restudy this at your own pace. But don't worry, you can also go to my YouTube channel, Biomolecule, and watch this video. Okay, guys? I guess, what time is it? Oh, 26. So reproduction in angiosperm is something you have to read. Yeah, we are going to discuss this when we meet again on Thursday. Please be sure that you read this part, okay, before you meet me. And I guess, okay. Oh, okay, it's just the, it's just the what? This is just the ending. So I guess that's it, everyone. That's what I got for you, for all of you today. Any question? Is there any question? Is there any question?